And I may have drawn this incorrectly. I have to admit I'm kind of spacing on whether we have in 5' overhang or 3' overhang, but maybe it'll come back to me. And then on this side, we have the BAM digest site and the bagel digest site. And then now you're probably thinking, well, BAMs can only ligate to BAMs, and bagels can only ligate to bagels. And generally, you're correct. But BAM and bagel are kind of a special case, and we have what's called compatible sticky ends. So a sticky end is this overhang that can base pair with other sticky ends. And if you look at the bagel site, it's A, G, A, T, C, T. And if you look at the BAM site, it's, let's see here, G, G, A, T, C, C. So we have kind of this area in the middle 4 base pairs are the same. And it just so happens that that is where our enzyme cuts. So it looks like, I think I actually did this backwards here. I think this should be sticking up, and this should be sticking down. So our BAM site is going to have a G, A, T, C site here. And then a bagel site is going to have an overhang with, let's see here, G, A, T, C. And you're probably asking, well, what about this extra C? You have to remember that was part of the other fragment of DNA, and that disappeared. So that's long gone. And if we look here, this G can bind to that G. Well, and again, I haven't reverse complemented. This would actually be C, T, A, G. But if we just kind of look at the homology, we have homology there. We have homology between these two A's, homology between these two T's and C's. So these are compatible sticky ends because the center 4, this sticky end, the center 4 base pairs of both of the restriction sites match up, and the outside nucleotides don't really participate in the base pairing. But what ends up happening is when this ligates, we're going to get a kind of a hybrid site that's half BAM, half bagel. So it starts out as a BAM, so we start to see a G, G, A, and then it still looks like a bagel or a BAM, and it could also be a bagel with T, C, let's see, I lost my count. Oh yeah, so TC, but then it turns into a bagel site with a TCT as opposed to a TCC. And this is neither a BAM site nor a bagel site, and this is what we call a scar sequence. So if we ever put BAM in here again, BAM can't digest this, bagel can't digest this. That means that this junction that we've put between our left part and our right part is permanent. We cannot digest that with anything. Furthermore, if we look at what this codes in amino acids, if we look at the genetic code, the GGA and the TCT codons give us a glycer. So this scar sequence is awesome in 2 ways. It's undigestible, it's like a one-way reaction, and we get a glycer linker, which is a great linker. So it's kind of a lot of information. Oops, looks like I need to fill out my chalkboard one more time. Let me do that. So I hope that makes sense, but I guess I'll try to do a quick summary and say that we have 2 plasmids. We have our left part and we have our right part. We have our eco, bagel, BAM, zo, eco, bagel, BAM, zo. And then for our left part, oops, let me change colors here. For our left part, we're going to cut there. For our right part, so this is BAM, zo. For our right part, we're going to be cutting bagel zo. And then we're going to get rid of these intervening chunks, however, whatever size they are. And then we're going to ligate these together. And so BAM and bagel, so that's BAM and bagel, are compatible sticky ends, which will then give us a scar sequence. So GGATCT, which also codes for a glycer. And so we get this coming together. So we get GGATCT. And then our zo digests also come together, so that in the end we end up with a plasmid that has L and R the rest of the plasmid. Here we have our scar sequence, which is a glycer linker. We have our eco, we have our bagel, we have our BAM, and we have our zo. We would then transform this into competent cells, either heat shock or electroporation. We would then plate it, we'd pick a colony, grow it up, mini prep it, and then map it. We would probably map it with eco and BAM to make sure everything looks the right size. And then we'd send out for sequencing with CA998 G00101 if we were, worried about mutation. Though usually if your starting parts have already been verified, the cloning process won't lead to any mutations. So if you're in a rush, you shouldn't wait for that. Let's see, a final note I would say is when you do your ligation, you transform, you get your colonies the next day, and you want to know right then and there, did your assembly work? At that point, you can go ahead and do a colony PCR right off of the colonies. So that if you have your plate, your colonies, what you do is you have, let's say I'll just make a grid. You have one part is your PCR mixture, your colony PCR mixture, and the other part is your media. And what you do is you pick a colony, and then you dunk it into the PCR mixture, and then you inoculate the media. And you do that for, 4 or 8 or 16 samples, and then you just run that PCR mixture, and it will tell you, if you run it with CA998 and G00101, like we do in our lab, it's just going to amplify out whatever is in between those two things, including your part. You can calculate this expected size. You run that on a gel. If you have the right fragment, then you know that the corresponding culture tube or culture well, if you're using 24-well blocks, is the right thing. And you can go ahead knowing that everything should work out. So that's assembly with a little bit of QC thrown in. If you have any questions or if I totally spaced on anything, let me know. And we'll be back soon with more.